Hi, I'm Charles, and today I'll be talking about cellular respiration. Cellular respiration happens in 4 easy but simple steps: glycolysis, pruvative oxidation, the Krebs cycle, and oxidative phosphorylation. There are 2 types of phosphorylation. There's oxidative, and there's also the substrate level phosphorylation, which takes place in the process of forming ATP by the physical addition of a phosphate group to ADP, and happens in the cytoplasm in the matrix during the Krebs cycle and glycolysis. So that substrate level phosphorylation happens during glycolysis, and the pruvative oxidation and Krebs cycle, which is, looking at the picture, glucose comes in, as glycolysis happens to start the process of glycolysis, which then goes into pruvative cycle right here, and then into the Krebs cycle. First, let's begin by discussing glycolysis. Glycolysis has four main ideas, which the first one is 2 ATP are needed to get this process started. 2. The breakdown of glucose occurs in cytoplasm. 3. This creates 3 carbon molecules. 4. The metabolic pathway of glycolysis is divergent, and each pruvic acid becomes an acetyl CoA and enters the cyclic pathway known as the Krebs cycle. Looking at the picture, let's go into a more descriptive process. Glycolysis happens in the cytoplasmic fluid of the cell. Glycolysis occurs by breaking down glucose into 2 molecules called pruvatives, which is right here. The glucose comes in, glycolysis creates 2 pruvatives, and the cell reduces 2 molecules of NAD+, forming the 2 molecules of NADH, and produces 4 ATP. So it comes in here, NADH to NAD+ to NADH, and then ATP. The next cycle, or process, is pruvative oxidation. Pruvative oxidation also has 4 main ideas or steps. Pruvative enter mitochondria if oxygen is present. 2. Loses a CO2 as a waste product. 3. NAD takes a hydrogen ion and 2 electrons. 4. A cell and coenzyme starts citric acid cycle. It will extract energy and electrons from organic materials. So looking at the picture, let's describe this process. A carbonyl group is removed from the pruvative and released as a carbon dioxide. So it comes in pruvative, and then carbon dioxide is released. A 2 carbon molecule from first step is oxidized, and NAD+ accepts electrons to form NADH. This oxidized 2 carbon molecule, or known as CO2 acetyl group, is attached to coenzyme A to form the acetyl CoA process. So this comes in glycolysis, and then pruvative NAD turns into NADH, and CO2 is released, and it forms this acetyl CoA process. The next process is the Krebs cycle. The Krebs cycle has 3 main ideas or steps. Pruvative acid is converted to acetyl CoA as it enters the mitochondria. Citric acid is the first molecule formed. Citric acid is broken down and new molecules are produced. Now let's go into a more descriptive process of that Krebs cycle, or the citric acid cycle, whatever you prefer. The Krebs cycle takes place in the mitochondria, so right here, TCA or citric acid cycle, or Krebs cycle, whatever you want to call it. Pruvative is transported into the mitochondria and loses carbon dioxide to form acetyl CoA, a 2 carbon molecule. When acetyl CoA is oxidized to carbon dioxide in this cycle, chemical energy is released and captured in the form of NADH, FADH2, and ATP. So it's saying that this process is used to create NADH, FADH2, and ATP. The next process is oxidative phosphorylation. Oxidative phosphorylation has 1 main idea or step, or overall general theme. The process by which ATP is formed as a result of transfer of electrons from NADH and FADH2 to O2 by a series of electron carriers. So let's go into a deeper understanding through our picture. Oxidative phosphorylation. The electron transport chain is a series of proteins and organic molecules found in the inner membrane of the mitochondria. So right here, the electron transport chain in the inner membrane. Electrons are passed from one member of the electron transport chain to another in a series of redox reactions. So redox reactions happen in the electron transport chain. Energy is released through these reactions and is captured as a proton granite, which is then used to make ATP in a process called chemolysis. So a protein granite is created right here to form ATP. Together, the electron transport chain and chemolysis make up oxidative phosphorylation. So those two things make this process of oxidation happen in the electron transport chain. These specific steps begin with the delivery of electrons by NADH right here. And then the next part is also that FADH2 is also used to begin this process. Reduced electron carriers from other steps at cellular respiration transfer their electrons to molecules near the beginning of the transport chain. In this process, they turn back into NAD plus and FAD, which can be used and released in other steps. The next step is electron transfer. The next step is electron transfer and protein pumping. As electrons are passed down the chain, right here, they form a higher to lower level releasing energy. Some of the energy is used to pump ions, making them out into the matrix or inner membrane. This pumping establishes an electron chemical granite. So right here. The next step is splitting of oxygen to form water. At the end of the electron transport chain, electrons are transferred to molecular oxygen, or O2, which splits in half and takes an H ion to form water, or H2O. So this forms carbon dioxide and water through using these chemicals or molecules, NADH, FADH2, and other ions on the electron transport chain. The last step is granite-driven of ATP synthesis. As H plus ions flow down the granite and back into the matrix, pass through an enzyme called ATP synthesis. So this whole process creates an ATP synthesis. So with these 4 simple steps and then descriptive details, that is the process of cellular respiration.